All right, so it is just about 6:35, so we can go ahead and get started. I just wanted to say again, thank you so much to everybody for being here with us. I know you had your choice of places to be tonight, including the school board meeting, so I'm pleased and impressed that you chose to be here with us at the library. A couple technical notes. Everyone is muted. I muted everyone when you came into the Zoom room. So if you do want to speak, you can either post something in the chat box that everyone can see, or you'll just need to remember to unmute yourself on Zoom before sharing anything. And there will be time for questions. Stephen has a presentation that's broken up into different segments. So there'll be a portion of each segment that is dedicated to a couple of questions. And then at the end, there will also be some additional time for questions. That said, I'm not sure. We can go ahead and get started. If it seems like things are a little bit choppy with the video or with Stephen sharing his screen or anything like that, we might ask you to just turn off your video. Sometimes that helps with the bandwidth. But for now, we've got about just shy of 40 people, so I think we'll probably be all right for now. But just a heads up that might be coming. So anyway, let us get right into it. I would like to introduce our speaker for this evening, Dr. Stephen Pelsue from Cape Elizabeth. He's the scientific director at BBI Solutions in Maine, which is formerly Maine Biotechnology Services, Inc. And BBI Solutions is a global leader in providing critical reagents and custom development for the in vitro diagnostic market. And I will not even pretend that I'm smart enough to know what all that means, but I'm guessing Stephen will give us the rundown on that and many other things. We're super excited to have him. So please welcome Stephen Pelsue, Dr. Stephen Pelsue.
Great. Thank you, Andy, and thank you everyone for taking some time tonight to participate in this presentation. I think I would recommend that everybody could turn off their video. It's been a little choppy on my side, just so we don't lose anything. I think that'd be great. Let's see, I'll go ahead and jump right in. All right, so tonight I plan to talk about the biology of COVID-19 more so than some of the other aspects of COVID-19 that you read about daily. And my goal was to put together a presentation that would give everybody a little bit more understanding of what's going on behind some of the decisions that are being made, thinking about public health issues, etc. I'm trying to make it so that it's accessible to everybody, but if something isn't clear, feel free to put a note in the chat. Andy's going to be monitoring that, and we'll take a couple breaks through the talk to be able to clarify some things, and at the end we can open it up for broader or more technical questions as people would like. I've broken the talk up really into four basic sections, and then the final sort of sections on thoughts about management and other aspects of how COVID is changing our thinking and our lives in general. So I'll go ahead and get right started. And I want to start out by just talking about coronavirus. So COVID-19 is actually the disease that is caused by the coronavirus that's referred to as the novel coronavirus 2019, or its official name is SARS-CoV-2. Mostly during the talk, I'll be referring to CoV-2, and that will be the designation for talking about the coronavirus that's associated with COVID-19. COVID-19 actually stands for coronavirus disease from 2019. And so when I say COVID-19, I'm talking about the disease. When I say CoV-2, I'm talking about the virus that causes the disease. And coronavirus is a large family of viruses. In fact, we've had 2 prior pandemics in the last 20 years that are coronavirus initiated. One is referred to as SARS-CoV, that happened in 2003, and really didn't impact the United States terribly. And there was some outbreaks in Toronto and a couple other places in North America, but it didn't really spread or distribute the same way that COVID-2 is. But they are related viruses, and they cause very similar types of clinical conditions, and therefore the present virus is referred to as SARS-CoV-2 to indicate that. There's another virus that very few people are aware of that's called MERS, which stands for Middle East Respiratory Syndrome. That's also a coronavirus, but much more distantly related. It's actually still quite active, but it seems to have been contained mostly in the Middle East. And of note is that there's not a lot of human-to-human transmission, but it's mostly animal to human, and so the outbreaks are less common and they don't spread nearly as wide. Of note, though, is that both SARS-CoV from 2003 and the MERS actually have higher fatality rates in general than the SARS-CoV-2, which may actually explain why we don't have the same kind of spread. Also,
is it known why some viruses are more easily passed between species rather than within a species?
Usually it has to do with interaction and how closely related they are to be able to infect the cells themselves. And so the transmission requires on a couple components. One is that you have to have very close contact to the animal host or the animal source, and it has to be transmittable in a way that humans can accept it. For example, MERS often arises from camel bites, not something that happens in Maine very often, and therefore you don't see a lot of virus transmission that way. So the other coronaviruses that are quite common are referred to as human coronavirus. And I don't particularly like that designation because it's a broad family, and all of them that I'm talking about actually are human coronaviruses with respect to their infection and transmission. But historically, we've called these human coronaviruses. They were the first human coronaviruses identified, and these are associated with the common cold. In fact, the estimate in North America is about 25% of common colds arise from the human coronavirus. And so the coronavirus as a family, this is not the first we've ever seen of it, both in terms of exposure, but also in terms of its spread and transmission. And we'll talk about that as we go through. So I want to talk a little bit about the biology of the coronavirus, but let's first start with just talking about what a virus is. And so a virus is essentially a packet of instructions. It's not considered a living entity because it does not have the ability to reproduce, which is one of the critical definitions of life, is the ability to reproduce itself. Viruses actually need to infect other cells and then hijack the machinery that they use to be able to produce the components to make more viruses. And as such, they essentially are a packet of instructions that enter or are delivered to a cell. And then that cell is taken over by that virus in such a way that the components, the proteins, the RNA or DNA genome that really are the blueprint of the instructions, get reproduced in the cell, repackaged, and then released from the cell. Now, essentially all living entities have viruses. So there are bacteria viruses, which is shown here on the right-hand side, and we refer to those as bacteriophages. And one interesting thing about the bacteriophage, which is one of the first very well-defined viruses that was studied is that they actually have this rocket ship-like appearance, and they attach itself to the bacterial cell and then physically inject the DNA into the bacteria for it to produce more. So this protein coat actually does not ever enter into the cell. That's quite different than the way animal viruses work, in which the animal virus is usually taken up entirely by the cell itself and utilizes some mechanism. We'll talk about that in a minute, to be able to enter into the cell, for which it then can co-opt the cell and utilize its machinery to replicate its necessary components. And so there are lots of different types of viruses. Several are diagrammed here. And one of the things that I want to point out is that there's sort of two broad families of viruses. There are DNA viruses and there are RNA viruses. And that has to do with the genetic material that the information is coded in. And if they're a DNA virus or an RNA virus, they utilize different pathways within the cell to be able to reproduce. And so that has more to do with how they're going to function inside of the cell than whether they're going to be more or less infectious. And then within those categories, you have some different structures that make viruses unique. And so most viruses have some kind of geometric structure that then has protrusions associated with it that are able to interact with the cells. And so typically these protrusions have some particular target that they bind to, and then that initiates them being taken into the cell. We'll look at this more specifically for coronavirus in a moment. And so these protrusions are actually what define what cells have the ability of being infected. And so when we talk about the virus host range, it's not only whether it's a human or a dog or a cat, but it's also within that host which specific cells get infected. And then that's going to have a lot to do with what kinds of symptoms or diseases can be caused by the virus. So the viruses also have sort of two different kinds of coats. And so broadly, they have either sort of a membrane coat, which comes out of the cell itself, and that has to do with how they're produced. The coronavirus actually falls into that category, as does the human immunodeficiency virus, whereas other types of viruses like adenovirus, for example, have more of a shell, a hard shell, which is made up of proteins that are tightly packed together, and it forms a protective layer. And so those have a lot to do with how long the cell can survive and in what conditions they can survive and how they actually interact with the target cells. So if we look at sort of a cross section of an average virus, we call this an animal virus, then what we can see is that there is some kind of protective coat. And again, this could have some membrane. It could be totally a protein shell, which would mean it would be sort of a harder, less penetrant type of structure. And then they usually have protective material that is associated with protecting the genome. So this is the DNA or the RNA that is the genetic blueprint for all the instructions necessary to make all of the proteins for it to form another virus. So the whole goal of a virus is to make more virus. So when they enter into a cell, they then co-opt a lot of different types of machinery and use the energy and the materials that are within the cell factory, if you will, to essentially generate many, many more copies of that virus and then release the virus back into the environment. And so the entire purpose or function of the virus is to make more virus. It can't do it by itself, so it has to find a suitable host. So if we look at the coronavirus by itself and think about the specifics of COVID-2, the virus that we're particularly interested in at this moment. We can see these are actually diagrams that are generated from the structures that have actually been produced by the virus. These bottom ones, which I'll get to in a moment, are actually three-dimensional structures of a particular type of protein, and then I was able to get that data and generate some of these images. These other images were actually generated by a group, I believe, at the University of Glasgow in Scotland. And what we see is we see that there are these protrusions on the outside, which I talked about a moment ago, that we refer to as the spike protein. This spike protein is a critically important protein for it to actually enter into cells. There is membrane, which actually comes from the host cell that it reproduced in. And so it doesn't make its own membrane. It actually steals it from the host. And then there are a few other types of proteins that can be on the cell surface, which are really more about maintaining the structure. It's a spherical structure in this case. The other major protein is something that's called the nucleoprotein, or N protein for short. And the nuclear protein basically covers the outside of the genetic material. And in this case, that's RNA. So COVID-2 is an RNA virus, and the nucleoprotein coats that entirely. This provides protection so it doesn't break apart or degrade so that it is no longer functional, but it also allows it to be packaged in a very, very small space. And so the nuclear protein is found inside of the virus, whereas the spike protein, or S protein, is found on the outside of the virus. And the reason why the spike protein is particularly important is the spike protein is what is going to interact with a specific receptor on the surface of cells in the human body. Okay, and we'll talk about this in a moment, or I'll show you a diagram. But the receptor that it binds to is called the ACE2 receptor. And the ACE2 receptor is found on lots of different types of cells, but predominantly it's found in the nasal passage, in the throat, in the lungs, in the kidneys, in the heart, all of the places where we see issues with coronavirus or COVID-19 disease. And so the spike protein will bind to that virus, and then that leads to its ability to be taken in by those particular cells. And what's sort of interesting is that the spike is actually made up of three identical proteins that come together in what we call a trimer. So the cyan and the green and the magenta are all individual units that come together and form this trimeric spike structure, and that you can also see on the surface up here. Now that spike structure is dynamic and has these little regions, which we refer to as the receptor binding domain, or RBD for short. And this is the part of the protein, this small little piece right here that's sticking up is the part of the protein that interacts with the human cell, that ACE2 receptor. That's the only piece right there that really is interacting with that receptor. And what's interesting is you can sort of see these three different units. There's one here, and then there's another one back there that you can't quite see. And these flip up and down sort of randomly. And then it has to be flipped up in order to interact with the receptor. And so what you see here is what we would call the open structure. And what you see over here is exactly the same structure, but it's in the closed structure. So the one on the left-hand side could interact with the receptor. The one on the right-hand side will not. So you can think of hundreds, if not thousands, of these on the surface of this virus, and they're sort of poking up and poking down. And so it has to interact in a very special way with the cell in order for it ultimately to be recognized or taken up by the cell itself. So let's talk a little bit about the infection process. But first, what I want to do is I want to sort of hone down a little bit more on the left-hand side. So if you focus on that part of the slide first, this is how the this is how the virus enters into the cell. So what we see here is we can see the virus diagrammed with these little spike protrusions that I just talked about. And then you can see the receptor, which is called the ACE2 receptor. So interestingly enough, the ACE2 receptor is associated with regulating blood pressure, but that really doesn't have as much to do with what's going on with the COVID-19, is that the cells that have this receptor are the cells that are then ultimately going to be targeted by the COVID-2 virus. Okay, so it binds to the receptor, or there's an interaction between the receptor and the spike protein. And remember, it has to have that little finger pointed up in order for it to interact. So if it wasn't pointed up, it sort of bounces off. But then another part of the virus could have one of the fingers pointed up, and then the receptor could latch onto it. And so it sort of will bounce around until it interacts appropriately. And once that happens, then essentially the virus melts into the cell. And there's a very interesting process by which this happens, but I didn't want to take too much time highlighting that piece. But I want you to just think about the fact that the virus sort of sits on top of the surface of the cell and then penetrates through the membrane into the cell. And so that's what we see here, is essentially the virus recognizes this particular target receptor, it melts into the cell, and then it opens up and is released into the contents of the cell. What happens then is that nucleoprotein that's bound to the genetic material is released from, in this case, the RNA, and then that forms the template for manufacturing more virus. So essentially, the life cycle of the virus is that it binds to the target of the cell, it gets taken into the cell, it makes all of the components necessary to build a new virus, and then it constructs a new virus, and then that virus is then released from the cell itself, okay? So again, the entire purpose or raison d'etre of the virus is really to make more virus. That's it. So it comes in the cell, makes more virus, and it spits more of the virus out to infect more cells, to then make more virus, to then spit more virus out. You get the picture. So what we see here is we see how that virus is then just routinely being produced. And one of the interesting things about this type of virus is that because the virus melts into the cell, and then we actually call this budding out in terms of how it gets released. And so you can think of a little bit of a bleb of the cell being formed, and then that just sort of pinching off like a little piece of dough and being released into the bloodstream or into the surrounding areas. And so what is interesting about that is that process doesn't kill the host cell. It may prevent the cell from functioning properly, or it may cause harm to the cell, but it doesn't actually kill the cell. So when I talked about the hard protein, the other type of shells that are found in viruses, those types of viruses make virus, but they don't bud out. What they do is they actually burst the cell open. So they actually make so much virus that it causes the cell to explode. And then it releases the virus out into the bloodstream or the surrounding space to then infect more cells. What that does, though, is that actually kills the cell that manufactured all of that previous material. So one by one, it begins to kill cells off, and that can actually be a limiting process, or a process of diminishing returns, because if it kills too many cells, then it can't make more virus. So those are sort of the two general functions. So one of the things to note is that this virus does not kill the cell, and that's pretty important. And we'll talk a little bit about that in a minute. So let's think about how it actually infects a person. So that's how it infects a cell. Now, there are lots of different cells in the body. There are lots of different types of cells, different tissues, and all of them have different proteins or different receptors or different components on the cell surface. But in this case, the only cells that are going to be infected by the coronavirus are the cells that contain this particular receptor, the ACE2 receptor. Okay. What's interesting is that that receptor is very, very highly expressed, meaning there are lots of these receptors on the nasal passageway in the upper respiratory tract. Okay. And so it appears that it is actually that nasal passageway and also leading down into the upper respiratory tract, although it really seems to be that nasal passageway that's really critical. And I'll talk about that in a moment, for the virus to then attach to those receptors and then be able to produce more virus and then release it. So that's stage one, is actually infecting the cells, and it appears that those first cells that are infected would be in the nasal passageway. And the reason why we think that is the case is these cells actually have almost 200 times more of that receptor on them than some of the other cells that we know get infected by the virus, which means that they're much more susceptible to binding to the virus and then taking it in. In addition, they seem to be very, very good at making the virus. So this causes two problems for us. One is it means that they're a really good target that once you breathe it in, both through your nose or your mouth actually, then those target cells can be infected by the virus. Okay. But then those cells are also really, really good at making more virus, and then they can spit out more of the virus. Well, that causes a problem because as you breathe it in, it now goes into your lungs. Or if you sneeze out or talk or breathe, then that actually releases virus into the environment. And so that's why the nasal passageway is such a problem for us in terms of the disease itself. So stage one would really be the target of the nasal passageway, producing more of the virus. And then as you breathe that in, then that will go into the lower respiratory tract, and in particular, the alveolar regions, which is where the blood exchange occurs. And we'll see that in a minute as well. And so what happens is that the virus will first infect here, you breathe it in, and then it will infect regions of the lung. And then the disease that actually occurs within the lung, so the SARS stands for severe acute respiratory disease. And so that severe disease is a result of the virus going crazy in the lung itself. In a couple different places, it actually can occur in the bloodstream and form clots. It can actually bind to specific cells inside of the lung, and then it prevents the passage of oxygen and CO2 across this barrier. So one of the purpose of these alveolar structures is for you to breathe in oxygen, and then that exchanges with the blood. And so the oxygen passes from here into the blood. The red blood cells take up the oxygen and then take it to the cells and tissues that need it. And then it will exchange the CO2 across this membrane. And then when you breathe out, you expel the CO2. And so now you have that blood exchange, oxygen and CO2 exchange. Well, what happens when the virus gets in here is it causes an immune response. We'll be talking more about that in a little bit, but that immune response essentially clogs up this system and prevents that oxygen exchange. And that's the acute respiratory disease. So it prevents the ability of oxygen to be able to be exchanged, which means that you can't breathe, okay? And so if we look at the timeline, the exposure is when we have that first association of the virus with the nasal passageway. And then the symptomatic early phase is when it makes it down into the lung itself. Now, there may be relatively little clinical or physical impacts at this stage, but if it goes on too long, and we'll talk about some of the things that can happen over here to make it a severe disease. If it can't get rid of the virus early on, then that's when we have the severe disease. And then that timeline is looking more like it's 7 to 10, if not more days than that, in terms of the first early signs of severe disease. And then unfortunately, it can just continue spiraling down from there. So now let's look a little bit more into this process and think more about the disease itself, okay? And so we have the cells being infected, we make more virus. That virus gets breathed in, and now we actually have it attacking cells in the lower part of the lung. And particularly, it's these alveolar sacs or alveolus that get infected. There's specific cells on the inside of the alveoli, and then also specific cells in the blood system that actually get infected as well. So it gets both sides of that pathway can get targeted. And so what happens is that in a healthy adult, as I've previously described, we have the gas exchange of oxygen out, and then the red blood cells pick it up and take it so that our brains and our muscles and every other cell in the body can function. And then it removes the CO2, which is a problem. And it's considered a waste product in this case. And the CO2 enters into here and then will exit when you exhale. And so these structures are really expanding and contracting, okay? And so they're very flexible. And part of what happens here is there's a surfactant which coats the lining, and so it reduces the surface tension, and so it can breathe like a bladder, okay? We'll keep that in mind for a minute. So now we have the SARS virus come in, and it begins to infect the cells both inside and outside. And then we start to drive a particularly robust or severe immune reaction to the virus. We'll talk more about what that immune reaction is, but essentially your body fighting the virus is one of the major problems that leads to the disease, okay? And so if the immune reaction generates substantial inflammation, then that can lead to the formation of fluid inside of the alveoli. It reduces the surfactant, and so the alveoli can actually compress and collapse. And then this fluid actually makes it so that the exchange does not occur from the red blood cells and the alveoli, so you don't get the oxygen in, you don't get the CO2 out. The CO2 then becomes toxic because it inhibits a lot of the same processes that oxygen actually enhances. And so as a result, you end up not being able to breathe, not because you physically can't breathe, but because the oxygen can't be exchanged in these areas. If this continues, then that's where we end up with a very severe disease. And unfortunately, if this is not rectified or corrected in any way, then this has the ability of leading to death. So this is the primary disease that we've all heard about. But I just want to mention a couple other things, is that the capillary beds, which are where you have these types of exchanges, but they occur in other places in the body that lead to different types of exchanges. This can actually prevent circulation. And so you may have seen in the news COVID toes. Well, that's one of the results is that these capillary beds get blocked up and that you have inflammation in those beds, and it leads to a pretty gnarly issue with the toes. The kidneys actually have a lot of this receptor, and we're starting now to see a number of kidney diseases. And then the heart also has a number of these these cells that contain these receptors, and so it can directly cause heart damage. And so there are a number of other target tissues which are equally as problematic and vital organs as the lung. And so once it gets into circulation, it can actually cause a wide variety of symptoms and create havoc that may or may not be controlled by the immune system, or even the ability of improving breathing by gas exchange and other things. Okay. So the last thing that I really want to mention before we can take a few questions about sort of the virus biology is that this exchange of virus interactions that occur here can lead to other kinds of side effects. So you may have read in the news that there can be strokes or clots that can be generated. And that's a result of the virus building up on these plaques. And so just like if you have too much cholesterol and you form plaques, and it can lead to breakage of those plaques, the same thing can happen, is that many of these same target cells are the same types of cells that are associated with plaque formation. So it can lead to blockage, or it can actually lead to the formation of clots, and that can lead to strokes or heart injury as well. And so there are a lot of different symptoms and clinical aspects to this disease that are associated with not only the virus infection, but what we're going to talk about next is really the immune response to the virus infection is what leads to the disease itself. Okay. So Andy, I'm happy to take a couple questions here if there are any in the chat that you want to summarize or clarifications. I'm happy to address this before we move on to the next section.
We do have one here from Joan and Arnie. Is there a medication to increase the surfactant? What role would nitrous oxides have?
So the surfactants, actually, those can actually be provided, and like newborns that can't breathe yet, and other people with different types of lung diseases, they often add surfactants to try and increase that surface tension so that breathing can be improved. Nitrous oxide has a number of different functions, both in terms of how it interacts and controls or regulates different aspects, as well as it can also change sort of the gas exchange pressures. And so there's a couple different ways that might be deployed.
And we have one here from Eric and Lori. Does the common cold coronavirus cause this kind of effect?
Oh, no, it does not. So the interesting thing about this, so there are multiple families and we classify them, and we're very, very clever in how we classify them, and we call one alpha and one beta. And so that grouping of the alpha coronaviruses are those coronaviruses that are associated with the common cold. The ones that we have seen in the pandemics are actually beta coronaviruses. So while they are very closely related, they are quite different in how they interact, their transmission. They actually interact with different receptors. For example, MERS interacts with a completely different receptor than SARS-CoV or SARS-CoV-2, as do most of the human coronaviruses. They do not lead to the severe acute respiratory disease or the SARS disease. They give you that common cold. And in fact, you likely have been exposed to many of these over your lifetime. And one of the things that's coming out in some of the data is the exposure to some of those may actually benefit us in helping us to fight off this particular virus. And we're starting to tease that apart a little bit. But I find that particularly interesting in terms of whether or not prior exposure to other coronaviruses have a beneficial or a negative effect on being able to fight the present coronavirus.
We have a couple more here. Can ACE inhibitor drugs affect the virus cell surface interaction?
So there's been a lot of examination of that, and I haven't seen any definitive data, although one of the targets that people are using for therapies is to try and block that interaction. And there's a number of different ways that that can be blocked. I haven't seen anything that has demonstrated that some of the ACE inhibitors, and actually it has to be ACE2. So ACE is a different receptor than ACE2, and a lot of the targets are ACE in terms of how the blood pressure is controlled. But there is some investigation and looking at a lot of these drugs and seeing whether they have any impact on viral uptake by the cell or regulating any part of that. But to my knowledge, none of them have shown a great promise as being therapeutics.
And would you like another question now, or do you want to move on and take more?
Let's move on, and then we can circle back to this. You know, people, please feel free to continue to type in those questions, and we'll try and address them all at the end if I don't get to ones that have happened here. But I want to make sure that I get through the material. So if you don't mind, I'd like to move on. So the next thing that I want to talk about is the immune defense, okay? And so let's first just talk about the immune system and how it functions and what its purpose is. So I think most people are pretty familiar with the immune system, and that it's really our defense system, and it is designed to protect the host. And every organism has some kind of defense system. We don't always call them immune systems, but even bacteria have defense systems. So believe it or not, they have the capacity of preventing those bacteriophage from entering into the cell, and they have specifically designed components to block that. And that would be the same kind of defense system that we might be using in a different way for the protection of animal viruses. And so every organism, every cell actually has some kind of defense mechanism. But when we talk about the immune system, we're really talking about a highly evolved, coordinated system that is used to protect against very complicated pathogens or infectious diseases. And the immune system itself is really a dispersed set of cells and tissues, and so it's not found in a single organ. So the picture you saw behind my head when we had the video on was an example of how the immune system organizes inside of a tissue. But that particular structure can be found in many, many different sites throughout the body. It's not just limited to one site like a pancreas, for example. And so as a result, it's a distributed system. And in fact, your skin is really the largest component of the immune system. And the reason is because it's a physical barrier. So anything that's a physical barrier, and what I like to say is there's the physical barrier of the skin, and then if you think of your gut lining and your throat and all of that, that's an internal physical barrier. And between those two are really what makes you you. And so the skin is designed to keep the outside out and the inside in and protect any kind of exchange between the two. And so everything that's a barrier, both physically or as a result of some kind of cellular response, is part of the immune defense system. But the immune system is really a organized set of cells, which we'll talk about in how they interact with one another to specifically target harmful proteins, viruses, pathogens, etc. Okay. And it's really one of the only systems that is really developing prenatally and continues all the way until you die. And so one of the things that fascinates me about the immune system, and one of the reasons why I studied it for so very long, is that you have this system that doesn't know what it's going to encounter over hopefully a 60, 70, 80, 90, 100-year period, but is really designed to protect you against anything that you could encounter. Okay. And so to me, how it's able to adapt and learn and identify what's good and what's bad has been one of the great fascinations of studying the immune system. But its essential purpose that I want to drill down on is to detect, destroy, and remove potential threats. Okay, that threat could be a virus, it could be a toxin, or it could even be a cancer cell. So the immune system actually can recognize your own cells and say that cell's going bad, we better get rid of it before it's a problem, and therefore prevent cancer before it starts. And so it really is designed to survey and protect. Okay. So let's look a little bit into how this happens. So this drawing that I put together, this you could think of as your skin or your gut. It's the epithelial lining, what I just said in terms of keeping the outside out and the inside in. That's the physical barrier. But there are times when that barrier gets broken. You fall and you scrape your knee, for example, or there are microbes, bacteria, viruses that can sort of squeeze their way in through the connections between these cells. So these aren't like cement, and therefore sometimes they can squeeze their way in. And so if they penetrate from the outside to the inside, then we need a way of actually alarming the immune system to be able to say, hey, there's a problem here. So actually signals get sent out whenever there's any kind of breach of the barrier. Then certain types of cells that we call phagocytes are recruited to that area. And what a phagocyte does, and what the name actually means, is that it consumes or it eats the bacteria or the virus or the pathogen. So their ability to essentially identify foreign material and consume it allows the ability of protecting from that material getting widely distributed in the body and causing harm. Unfortunately, that process by itself is not enough to be able to completely rid your body of specific harmful elements. And in addition, a lot of these harmful elements have designed ways to try and avoid being recognized. And so there's a constant battle going on there between your immune system and these crafty little microbes. And so a second stage that can happen is that these get consumed by certain types of cells, and then they get taken into areas where we have cells of the adaptive system. These are the cells that make the antibodies, so the B cells and the T cells. So the B cells are the cells that produce the antibody, and then T cells have two different functions, one of them helping B cells make antibody, and one actually targeting cells that are a problem. And I'll talk about that in a moment. So this is really an orchestrated event that then allows the recruitment of these pathogens to the specific target. And this would be one of those areas where you would see that nice little green and blue. They're not really colored green and blue, but or red, I guess it was. But this is the area where you would see those structures and where these cells will interact. And so antibodies get specifically designed to recognize this unique organism. Okay, so the cells here, these phagocytes, we call them innate cells, or the innate response, will recognize essentially any foreign material. Okay, and it won't discriminate one foreign material from another. The antibodies in the T cells actually recognize very, very unique components of that so that it knows exactly what that bacteria is. So you could have two different types of bacteria. These cells would recognize both types of bacteria, whereas the antibodies or the T cells would only recognize one of the type that it was targeted to. This is beneficial because what it does is it improves our ability to capture and detect and remove and block from future infection. And so what it does is it makes the immune system specific. And the other thing that it does is it generates memory. So after you've had that response, there are cells that we call memory cells that can be reactivated if that infection comes back. Okay, and that's the whole purpose of a vaccine, is developing memory so that we can fight a response. Okay, so this whole pathway is a really organized and coordinated function that allows us to have a general recognition of potential threats and then very specific recognition of potential threats. So let's think about the virus specifically as the threat. Okay, obviously coronavirus falls into this category. So there's a couple problems with viruses in terms of the immune response. So one of the things that viruses can do is can get inside of a cell. Okay, once they get inside of the cell, they actually can be hidden from the immune system. And what that means is antibodies can't get inside the cell. So if the virus runs inside the cell and shuts the door, then the antibody can't get in, can't identify it, can't remove it. Okay, so the ability to actually get hidden inside of that cell is one of the mechanisms that pathogens can use to try and protect themselves against the wrath of the immune system. So we have two different arms or branches of the immune response. I mentioned these a moment ago. The innate response is really just the ability of generally recognizing any kind of threat. And it doesn't really have memory, so it doesn't really change the first time it sees something versus the second time it sees something. The adaptive immune response is the response that generates a very, very specific response to that particular pathogen. In which case, the first time it responds, it might be slow, but catch up and then help identify and detect it and remove it. But then the second time it sees that, it will activate very, very rapidly and clear out the virus or the pathogen before it becomes a problem. Okay, so that adaptive response has this memory capability that improves the specificity and improves how the immune system can fight this off. So a virus would be recognized sort of generally by the innate response, and it actually sends out signals. Okay, and we call these molecules cytokines because they basically are how one cell communicates to another, is they send these signals to each other. And so you can think of it as the telephone system, is that they send these signals, probably more like Morse code actually, that they send these signals, and then the cells that are geared to respond to those signals then get attracted to them, and then that leads to the deriving of the response. So these are referred to as cytokines or interleukins. So you can think of these as just really communicating from one cell to another. So these cells sort of throw up the flag and say something's wrong. These cells, the B cells or the adaptive response, comes in and it adapts. It actually learns what that response needs to be, and then it generates a response to be able to fight it off. And there's a couple different forms of that. There's an antibody response that is often referred to as a humoral response. So this is really a soluble response, so something in the bloodstream or something that is out in circulation. And then there's a cellular response, and what this response is a different type of T cell that we call a cytotoxic T cell, or referred to frequently as a CD8 T cell. Probably more information on T cells than you wanted to know. But these types of T cells actually will recognize the fact that there's a virus inside of that cell and then kill that cell. So that cell takes one for the team. So if you kill the cell, you kill the ability of the virus from being able to produce more virus, and therefore you essentially prevent the spread. And so if there's a cell that is infected, then these CTLs, the cytotoxic T cells, will then target that cell and then kill it off. This is actually the same way that our own body helps fight cancer, is it recognizes this is a cell that's gone bad. We need to get rid of it before it becomes a problem. It's the same type of response, whether it's a virus, whether it's a cancer cell, whether it's a pathogen. And sometimes we have a response that occurs where it recognizes ourself and it shouldn't. And those are referred to as autoimmune responses. So diseases like lupus or rheumatoid arthritis or multiple sclerosis are a result of the immune system recognizing our own cells rather than harmful cells. So it's got to learn what's a problem and what's not, and sometimes it gets confused and can cause other types of problems. So in this case, the coronavirus would penetrate into the nasal passageway, produce more virus, and so the first response would be this innate response, recognizing both the cells, because they're going to send up flags saying, hey, something's going on here, and then you get all of these types of cells come into the nasal passageway. Well, whenever you get sick, when you get a cold in your nasal passageway, you generate a lot of snot. Well, that is actually your immune system forming a viscous layer in order to prevent the virus from escaping that area. And also it's the result of cells being killed off in trying to fight that virus. So as a result, that snottiness is really part of what happens as you try and fight the disease. But then they also then bring into play the adaptive immune response to then be able to specifically recognize and target that one component, and that one component then becomes much more efficiently recognized and removed. Okay. So let's think about this with the response to COVID-2 specifically. So it's a virus, it gets inside of the cell, and so we really need both the innate response to be able to recognize the virus, and then we call it presenting, but it actually recruits and interacts with the adaptive immune response. And so it's really a very orchestrated interaction between all of these different cells, and they have unique functions, and they need to function together in a proper way. So we have an infected cell, and then that CTL, that killer T cell, will recognize that, and then it has the ability of killing the infected cell, which hopefully will then shut down virus production by that cell itself. Now, what antibodies are doing is they actually can bind to the surface of the virus, and then that helps them being picked up by other types of phagocytes and therefore removing the virus, but it also can block the virus from actually penetrating the target cells. So if an antibody is bound to that spike protein and prevents it from interacting with the ACE2 receptor, then the virus can't get into the cell. And so even though that virus is present, we've sequestered it and therefore removed it from being a problem. We call that neutralizing. And so some of the therapeutics that are being designed are actually to try and develop antibodies that we can inject into people that would block the ability of the virus getting into cells, and therefore it substantially would reduce the spread of the virus. Okay. And so both of these arms of the immune system have to be active. So both the innate response, the adaptive response, and then both branches of the adaptive response, the antibody response and the cytotoxic T cell response, all have to be activated, but they have to be properly coordinated. Okay? So this is where the immune system actually can cause the COVID disease. All right. So the virus itself infects the target cells. But what really drives the disease is the inflammation from how the body and how the immune system responds to the virus infecting those cells. So all those branches, so the innate response over here, the adaptive response over here, and then another part of the response, which is actually sort of helping to control and mediate that, all have to interact properly. And remember, these cytokines get sent out and help to organize and bring the right cells and drive the right response, okay? And so we need this to be recognized, drive an antibody response, and then drive a particular cellular response to coordinate all of this. Now, what's interesting is the type of cytokines that get produced are basically in response to the type of pathogen. Is it a bacteria? Does it get inside the cell? Is it a parasite? A number of different factors will lead to different types of cytokines being produced. And that will regulate the type of response that's necessary to be able to fight off that particular disease. Okay? What is very interesting about the coronavirus 2, COVID-2, and the COVID disease is that what seems to happen for people who get very severe disease is that the cytokines that get produced are cytokines that are really targeted towards recognizing a different type of disease, meaning a different type of pathogen. And because that pathogen isn't present, they keep producing more and more and more of those cytokines. We refer to that, and you may have seen it in the news, called the cytokine storm. The problem with the cytokine storm is it overwhelms the immune system and generates this huge inflammatory response. We call that hyperimmunity. And so what happens is that you basically drive the immune system to respond in such a way that it doesn't actually kill the virus, but it actually causes damage to the target tissues. Now, the immune system should be able to identify uniquely the aspects that it can target and then surgically remove that without damaging the host. What happens in this case is it sends out the wrong signals, so the immune system gets very overactive with this cytokine storm, and the inflammatory response becomes overwhelmed and doesn't shut down. And then that causes the damage to the organs, particularly in the lungs. And so this inflammatory response leads to fluid in the alveoli. It leads to damaging and killing off of some of the cells that are associated with the gas exchange. So as a result of the virus infecting, it's really your immune system or your body trying to fight that virus that causes the COVID disease. Okay? And so what we have to do to try and block that is actually regulate the immune system or block parts of the virus that don't activate it. Now, people who have very mild disease, or some of those that we call non-responders, probably coordinate this response very, very well, and therefore don't generate that substantial inflammatory response, and so they don't have symptoms. Okay? And they may be able to remove the virus very, very quickly, and so they don't even spread the virus very much. Okay, so the spectrum of response that we're seeing often is associated with the spectrum of response that we see our immune system trying to fight that off. So this is another opportunity, I guess, to answer a couple questions around the immune response. Then we'll move on to testing and then sort of wrap things up after that.
All right, so let's see, going backwards here. Jessica had a question. Why or how do some people only get a slight version of the disease? Why isn't the virus doing its thing to make more virus in these people?
So we're starting to learn more about that, and there's some really interesting recent publications that have looked at what we call fingerprinting this immune response. And what we're seeing is that early on, how the immune system starts to respond may actually be a predictor as to whether they get severe disease or even die, versus those that get mild disease and then recover from it, or those that don't get the disease. So there's a few different things. So it has to do with how your immune system responds, and a lot of that may have to do with things that you've been exposed to in your lifetime. Many of the things may be associated with being male versus female. So males have a higher infection rate than females, and it turns out that most males have higher levels of ACE2 and many of these target tissues. And some of it may have to do with how your immune responds very, very early in that process that can set it up as to whether it becomes a very severe disease or not. So there's some really interesting data that's starting to come out that is beginning to give us some insight in terms of why there's such a wide variety of responses. But all I can tell you right now is that it seems to be the individual and how they respond as to whether they have no symptoms, mild symptoms, or severe symptoms.
We have another one here from Joan and Arnie. Now that we know more about the progress of this virus, what are the newer ways we may intervene at an earlier stage in the treatment protocol?
So there's a number of different things that are coming out. You probably have heard about remdesivir, which is a therapeutic that actually inhibits the virus from being able to replicate. And so it actually stops it from being able to produce more virus. Its effectiveness is variable, and so it's not going to work for everybody, but there's sort of a class of those kinds of drugs that people are looking at to try and block the virus replication. Similar to what was done with HIV, and that being able to block the virus's ability to produce more virus is really the way that the disease was substantially prevented from causing further damage to people who were infected. So that's one approach. There are a lot of different antibodies, therapeutic antibodies. So they're making antibodies that target the surface, particularly the spike protein, that then can help the immune system clean that up. And so if you inject those very early on, then that might prevent further infection and therefore limit the spread from that person to other people, but also within the body. And then there are a number of other things that are really targeting things like the cytokine storm, trying to shut down that storm. So you probably heard in the news that corticosteroids had a dramatic effect. Well, what corticosteroids do is essentially shut down inflammation, which means it shuts down this signaling. So it's a huge hammer that it's whacking and shutting it down all over the place. But by doing so, it may limit the ability of that cytokine storm and that massive inflammatory response from happening. And so there's a number of different fronts that people are trying to, again, it's really going to be a number of different therapies necessary at different stages to try and address different problems.
And there are definitely more. Would you like to come back to those at the end?
Yes, please. Yes, please. So we've talked about the sort of the biology of the virus and how it infects us. We've talked a little bit about how we try and prevent viruses, and particularly coronavirus, from causing problems, or what happens when it does cause problems. So the next thing that I want to talk about is really about testing. And so one of the key things that we have, because we don't have a therapy, we don't have a vaccine, talk about that in a little bit, but is to be able to accurately test people. So I want to talk about testing a little bit. And this is actually changing quite rapidly in the last week or so. So let's talk about testing. Screening is when you're actually testing for someone who is asymptomatic to try and determine if they're a potential risk. Okay. Whereas a diagnostic testing is somebody who has symptoms or believed to have a particular condition, and then you're trying to specifically identify if that person has a specific type of condition. Okay, so these that's sort of general for medical testing across. So an example would be a mammogram or a PSA. A lot of people get these exams even though they don't have any symptoms, to identify whether they should be further examined or to be able to provide baseline. So if they do demonstrate some kind of symptomology, that they could be tracked. Okay, but let's say that you then have evidence, because you felt a lump, that there was a potential risk of having cancer, breast cancer. Then you would do a biopsy. So the biopsy would be diagnostic because it's specifically looking for cancer in that tissue, whereas a mammogram is just looking to see whether or not there's any potential there, hopefully catching it early. So even though you're doing sort of the same type of testing with regard to evaluation, they have different purposes and different criteria. For example, screening might have a very high false positive rate, meaning that it may identify people who really don't have it as a flag that should potentially be evaluated. The PSA is prone to a very high false positive rate. Okay, but that's okay, because then you take a more detailed look at that and you can distinguish whether that person actually has prostate cancer. So it's throwing a big net out there. Okay, and trying to capture everybody and identify that. Diagnostic testing, on the other hand, needs to have a very high specificity rate, meaning that it has to have a very low false negative rate, meaning that it only identifies people who have that condition. Okay, so you don't want to have a false negative, meaning that somebody doesn't have it and therefore not treat them. And so the two have sort of different criteria in how we would evaluate that. So there's just a little bit of background on sort of types of different kinds of testing. So how about testing for coronavirus? So there are really three different types of tests that are out there in terms of the categories, okay? And those are on the left-hand side of the slide here. There's the molecular test, and this is actually testing for the genome of the virus itself. And this actually can amplify that genome a large number of times so that you can have a very small quantity of the virus present and still be able to identify that, okay? And so this molecular test is really to identify if you are infected, okay? If the virus is not present, so let's say you had the virus and then you recovered from that and there's no virus present, if you did this test, you should show up negative. However, if you did the test while you had symptoms or after you have been infected and are starting to produce more and more of the virus. So now you can see why those nasal swabs are one of the ways that people are testing for the virus, because the nasal area is one of the initial infected areas. And so that area could have virus in it before you show any kind of symptoms. Now there's actually a saliva test that's doing a very similar thing, but it's using the same basis. That area has the earliest infection rate and produces a lot of virus. And so by swabbing or spitting or analyzing that kind of information, you have the ability of detecting it very early on, okay, before you may show symptoms, okay? So that's one of the differences, and so that would actually be a screening test. But it also could be diagnostic. If you show symptoms, then you'd have that test to confirm it, okay? Now, an antibody test is testing for if you make antibodies to the virus. So you essentially are a later stage in the process. So antibodies typically don't show up until 10 to 15 days after you've been infected. Okay, so you have the ability of detecting using the molecular test if you have virus present, probably within a day or two of being infected. However, you can't test whether you've made antibodies until about 10 days later. And if you remember, when we looked at the timing of the disease, 10 to 14 days and beyond is when you might actually have severe disease. So the antibody test isn't a good test to diagnose whether you are infected. It is a good test to determine if you have been exposed and don't presently have symptoms. So if you've recovered from it, you didn't have a test when you were infected, then having an antibody test means that if you have antibodies present, the virus was present but may not be present now. And therefore it can identify people who have been infected but may not have any symptoms or may not even have the virus present. Okay, so those are sort of the two general types of tests that you probably heard in the news. And for those people in the Maine area, one of the most widely used molecular tests was developed right here in Scarborough, Maine at Abbott Laboratories. And so that's one of the major high throughput tests for evaluating the presence of virus. The antigen test is a test to try and identify whether the virus is present. So this is the reverse of the antibody test. It actually uses antibodies that we make. The company that I work for, we actually are making some of these antibodies to then be able to identify and capture the virus from samples, okay, blood or saliva, although it's not present in blood very much. And then this would identify whether the virus is present, but that test can only pick up when you have larger amounts of virus. So the molecular test and the antigen test are serving the same purpose of identifying whether the virus is present and therefore you're infected with the virus. But the molecular test can pick it up much earlier than the antigen test can. Although now what we're seeing, and this is one of the new sort of developments that's coming through, is some of these antigen tests can actually be converted to home test kinds of kits, whereas the molecular tests can't. So the hope is that we could come up with a test that would be useful, but then could be very widely distributed, very easily used, and then be able to screen people on a much wider basis than we presently can. And so all of these tests are being generated. There's literally hundreds of different tests out there. And one of the things that we're learning through these tests are what are the things that we really need to detect, both in terms of early recognition so that we can isolate a person, as well as identify those who may have been exposed to it or are presently actively exposed to it. So these are the types of tests that are being developed and deployed today. The company that I work for, called BBI Solutions, actually was part of a consortium that is developing an at-home blood test for the antibody test. And this was actually developed for the UK government to be able to have a mass test to be distributed through the UK. There are actually similar efforts going on in the US. Our company, whose headquarters is in Wales, was contracted to help with the development and do a lot of the manufacturing of this test, which is now about to be released. And so some of these tests are starting to be developed that are sort of what we call over-the-counter or point-of-care kinds of tests, so that it makes testing easier, doesn't require very expensive equipment and so forth. So we're making a number of different progress steps to be able to hopefully increase testing. And this is obviously a global goal. So a little plug for the company there. I'm going to go right into vaccine, and then we'll be able to sort of take a number of tests, because a number of questions. Sorry, there's no test, and then we can sort of wrap up, and I can take questions, open it up more broadly. So you've obviously heard a lot about vaccine development, and so I want to talk a little bit about what vaccine development looks like, and then some of the vaccines and where we are in that process right now. So vaccines essentially prime the immune system to recognize a potential threat without actually ever having seen it. So we prepare something that doesn't cause the disease, but then primes the immune system. So then when you get exposed to what we would call the native pathogen, or the pathogen that you would get exposed to in the environment, then your immune system would be able to respond very quickly, and then you would not get as sick, or maybe not even sick at all, okay? And so there are a few different types of vaccines. There's the live attenuated vaccine, and this is where they've actually modified the pathogen so that it is a live organism or a live virus, meaning that it has the ability of infecting cells, but they've removed parts of it so that it doesn't cause the disease. So an example of this is the measles, mumps, and rubella. This is an attenuated vaccine. And so essentially it acts almost identically to the native exposure, but at the same time, it does not cause the severe disease that you can get from measles in particular, which is a very contagious disease. And therefore, it primes your immune system very, very strongly to be able to respond to that if you encounter it normally. Now, not all things can be attenuated. There's a very specific process that you have to do, and you have to make sure that it can't revert back to its wild. And so it's not an easy process to develop these, but this is one strategy. An inactivated vaccine means that the pathogen has been killed. And there's chemical and a number of different ways that they can kill the virus, so it no longer has the ability of replicating. It will infect cells by being taken in, but it won't actually replicate, won't cause the symptoms, but really primes your immune system to develop it, and it's using native material. And so the flu vaccines that you get are often of this type, and then the polio vaccine, of which there are a number of different polio vaccines, but a majority of them were vaccines that were inactivated. If they take pieces of the virus or the bacteria and then they use that as the vaccine, then that directs the immune system to a specific component. So it's not putting any live virus, attenuated or inactivated. It's just adding a small part of that we know will drive an immune response that will then be protective when it does come in contact with other native exposures. And so, for example, the shingles or the new HPV vaccines, which actually have lots of different components to them to prevent against a number of different HPV exposures. And then the toxoid vaccines, because for like tetanus, for example, or diphtheria, they actually produce toxoids or compounds that themselves lead to you could think of them as a poison. Many of them attack nerves or other things. It's not the bacteria itself that's really the problem. It's a product that bacteria develops and therefore causes specific problems. And so the toxoid vaccines are typically to try and capture that toxin so that it can remove it and therefore doesn't cause the problems. Okay, so these are sort of the different kinds of vaccines that historically have been developed. So let's look at how you go about developing a vaccine. So let's look at the top part of that. And what I want to do is I want to sort of break down the process. So the first part of the process here is really identifying what the target is and doing the research to be able to know how you can develop a vaccine, what you need, what kind of immune response needs to be generated, what components need to be blocked or targeted in order for it to be protective. Okay, and so this is all the research that goes into understanding how you would design a vaccine. So then you've done that, which literally could be years and years worth of work. And then you would start your first clinical trial. So clinical trials in humans are broken down into 3 phases, and then there's the licensure or the actual development or release of that drug for a specific purpose. So let's just break down the phases. The first phase is where you're testing it in humans to make sure there aren't any sort of side effects. Okay, so this is actually taking healthy individuals, giving them different doses and seeing if there are any side effects. And so this is done with a small number of people in a very controlled way. And then they're measuring a lot of different things that are really related to making sure that the vaccine that they are producing are safe and don't cause unexpected problems. So then if that moves forward, and then you have to report this data out and then get the approval to go on to phase 2. Then phase 2 is where you then take a small number of people that begin to ask whether this vaccine will actually do what it's supposed to do. So in the case of a vaccine, does it develop an immune response? And does that immune response actually protect against, let's say, the virus? And then if that is the case, then you go to phase 3. And so phase 3 is a very large trial. So you may have read in the papers, there's a number of different vaccines that have moved into phase 3. Talk about that on the next slide. But that phase is where you might look at 30,000 or 50,000 people and then either naturally let them get exposed. So do the people who have the vaccine versus the people who have the placebo have a much lower rate of getting the flu, for example, or that kind of thing. And so this is a very generally a longer study than any of the other phases, and it's much, much larger. So in the case of vaccines, we're typically talking about 10 to 50,000 people need to actually be treated in phase 3 to understand that. And then all of that data is reviewed, and if the phase 3 shows appropriate, what we call efficacy, meaning that it works with very low side effects, then it can be licensed. Now, the efficacy or the rate at which it is effective can be different for different things. And so the flu vaccine often may only have an efficacy of about 30%, but there are several different flu strains in one vaccine, and therefore each of those strains may have different impacts. Whereas a vaccine for, like, let's say MMR needs to have a very, very high efficacy rate because it's for a lifetime of exposure as opposed to on a seasonal basis. So those different types of processes. So all of these are essentially the various steps that you would go through to develop a vaccine. Now, the things that you have to think about from the company side of things is that for these first two phases, you're using relatively small amounts of the material, whereas the third phase and beyond, you have to be able to manufacture it. So you have to be able to manufacture it large scale, meaning generating the number of doses that are necessary to treat the population. But in addition to that, you have to be able to make the same thing time after time. And then in addition to that, you have to be able to make it so that there aren't contaminants or other pieces in there that could cause problems. So this manufacturing scale up in and of itself is a very, very difficult thing. Okay, so that's a traditional program. And if you look down here, here's some examples. This is actually a figure from a New York Times article a month or two ago. And it shows the number of years that it took to get a chickenpox or a flu vaccine or the human papilloma, the HPV virus, and so forth. And you can see that we're talking about 15 to 30 years that it could take to generate a virus. I will tell you that these two here that took so long are really using different older technologies, where some of these other ones are newer technologies. And so I would say if you were to start today in a normal process, you're probably expecting a 10 to 15 year time frame in order to be able to generate that vaccine. Okay. So in a pandemic, you can't wait 10 or 15 years. And so the goal here is to develop a vaccine in 18 months. I think we'll not necessarily have that delivered, but I think there will be some identified in that time frame, which is a remarkable thing, by the way. Just to put that in context, the fastest we've ever generated a vaccine in North America, and I think Europe as well, is 4 years is the record. So you can see that this is really a lofty goal. Well, how do we do that? How do we take a process that is so long for good reason, and then compress it so that we can shorten that? So the first thing is to actually begin to overlap and compress some of these sections. So the clinical safety and dose selection get combined from phase 1 and phase 2, and then actually efficacy after that. And so basically you start to blend some of the phases together to be able to use the same data set for multiple purposes, and then evaluate the data at the key time points to make sure that it still is meeting the goals of the program, still is safe, etc., but you're compressing that. And that requires huge effort, because it means that the moment the data is available, it has to be reviewed so that you can move to the next step. And so organizing and coordinating this is a very, very difficult thing to do, from data generation to presentation to review to decision making. The other thing is that what we're seeing now is we're actually seeing huge investments in the scale up in the manufacturing process happen way before we actually have the vaccines done. Bill and Melinda Gates, for example, have invested hundreds of millions of dollars to build 7 different factories across the globe to be able to scale up this. And a lot of contracts have been done by the US, by Canada, by the UK, by European Union, to actually contract companies in advance before we even have a vaccine and get them prepared to be able to produce the vaccine. So the scaling and the manufacturing side of that is happening far, far earlier in the process. The third piece of this, which allows this to move forward, is we actually had a lot of research already in place that allowed early development of potential vaccines. So because we had the pandemic in 2003, we had the pandemic in 2012, we already had and were working on different vaccines for those. And so basically the coronavirus 2 components are being swapped into those models based on what we knew, because they're so similar, so that we had a head start. We didn't have to start from scratch on the research. And so because we had that information, we had a jump start on what we would do for targets, and then they were moved through quickly. And so all of those things have led us to be able to compress this and hopefully get a vaccine much, much sooner than you would normally generate. So let's look at where we are with the coronavirus vaccine in general. So this is the New York Times actually has a live virus vaccine tracker. Tells you what phase, how many there are, and gives you information about what they are. I'll highlight this and say there are 2 that are approved. One of those is in Russia. It never went through any kind of phase 3 type trial, so we have no idea how safe it is, whether it works or not. So I wouldn't rush out to sign up for that vaccine. There's also one in China that also is sort of questionable with regard to what kind of safety pieces it put into play. So while there are 2 that are approved, they're not widely available, nor would I recommend you run out and get them. However, there are 8 of them. I've highlighted a few of them over here that are showing real promise and are in the early to late stages of phase 3, meaning that they're very promising at this point. And many of these you probably have read about or seen in the news. So Moderna and BioNTech. So Moderna is in Cambridge, Mass, and BioNTech is actually in Germany. These are both using what we call mRNA vaccines. And so this is a coding material that will generate proteins of the virus. It won't be replicated, but it basically will be delivered to a cell and produce components of the virus inside the cell, so it will look very much like a virally infected cell and generate immune responses to those specific targets. It doesn't generate the virus, it generates components of the virus that we know the immune system has to respond to. And so these 2 are both looking quite promising in terms of the data they've generated. The CanSino Bio, which is a Chinese company, and the University of Oxford in England are using an adenovirus delivery system. So adenovirus is a different type of virus, and they've modified viruses. The one the University of Oxford is using is actually a chimpanzee version that they've modified. And what they're doing is they've essentially taken these viruses and added components to the virus that are coronavirus components. So it doesn't cause any kind of disease as a result of the adenovirus exposure, but it's presenting components of the coronavirus to the immune system, just like if they were virally infected. And so the University of Oxford one is looking very promising. And I'd say the top 2 at the moment are probably Moderna and Oxford in terms of the amount of data we have and the predictions of where they're going. All of these, because they've made it this far, are also looking pretty good. The Wuhan Institute in China and Sinopharm, which is the Chinese government-owned manufacturing component, are using inactivated virus, and they're testing these. And there's a couple other strategies that are being used. These are the ones that are most highly visible in terms of data and what we know, and have the highest likelihood. So one of the things that I'll say is that some of the criteria that's being put forward to determine if they are effective is that they have to be 50% effective. So it doesn't have to be 100% effective, but it can't be less than 50% effective in order to be considered. And I think it's quite likely we're going to have different vaccines that are going to have different efficacy in different people. And so we need a number of different viruses so that we can treat it both globally, but even within different populations. So I'm hopeful that a number of these go forward and are all promising, so that there's really the ability of fighting it in a large way. The other thing is that, for example, the U.S. government has already contracted with, so these companies are partnering, or these universities are partnering with manufacturing companies for them to make it. And so most governments, the UK, Canada, the US, European Union, are essentially contracting these production companies to make a certain number of doses. For example, the US, I know, has recently contracted 100 million doses from Moderna, and I believe 100 million doses of, I can't remember the company that's manufacturing this one, but from this as well. And I think the BioNTech has a contract, the US. Other people are contracting different pieces, but we've already started to contract these doses. You would never see that in sort of a non-pandemic way. The government would wait to see how effective it is and which ones work before it would even contract for production of these. So that's one of the ways that we can also advance this forward. So the last thing I really want to talk about is something that's called herd immunity. You've probably heard about it, read about it, etc. But I want to explain it in very briefly, because a lot of people have a misconception of herd immunity. A lot of people think of herd immunity as culling the herd, and it's actually not that. It's actually generating enough immunity within the herd or within the community so that it prevents the spread. So if you cull the herd, then you're left with just susceptible people, which means it makes it more infective within the people who have not been exposed or who are susceptible and, for example, couldn't get a vaccine. And so in those contexts, if you cull the herd, you actually may threaten the herd to a greater extent. Whereas a herd immunity means that if you can protect enough people, and it doesn't mean 100% of the people, but if you can protect enough people, that it limits the spread. So in this diagram, if we start in the upper left and then go through this, we can see if nobody is protected, that when one person gets infected, they can then transmit it to anybody who is not been exposed before, doesn't have immunity to it, and then certainly that could spread wildly within that population. But the more people that get exposed to this and then have immunity means that when the infection comes in, there is no way that it can be transmitted, or the transmission is reduced or blocked. And so herd immunity is shown down here. So the people in sort of yellow would have immunity, people in blue would not. So the person that's infected would have very limited transmission. And so what that does is that blocks the ability of the virus to spread through a community. That doesn't mean that the virus absolutely goes away. It just means that the community spread is shut down. This is what vaccines are designed to do, is provide that herd immunity. So if it comes into a community, that spread is dramatically reduced, and so fewer people are infected. The goal is obviously that nobody beyond the initial person getting infected gets transmitted to, but that's a very difficult goal in any circumstance. Now, the predicted population, and this is going to vary in different areas, so this is really community oriented, but the estimations are anywhere from 30% to 70% of the population, and I think those are in different communities, and the prediction that goes into that, it would be a whole another lecture. But so the number of people that need to have the vaccine and be protected probably is somewhere in the 40 to 50% range in order to prevent transmission at the rates that we're seeing it today. And so that's the goal of the vaccine, is to essentially generate herd immunity without harming the population. If you allow natural herd immunity to be generate, you actually put a lot of people at risk. A lot of people will die. A lot of people will have long-term impacts, which is why the vaccine is really so critical. So just the last sort of summary piece is, where do we go from here? And I saw this in a review. Jamie King is a lawyer, a biological biopharma kind of lawyer at the University of California in San Francisco, I believe. Wrote a really interesting article, and had this quote that I find to be really poignant in what we're dealing with. And so the management right now is really around preventing the spread, because we don't have therapies and we don't have a vaccine. And so it's preventing the spread by distancing and mask, testing as much as we possibly can, and isolating people until we have a vaccine. And so then the strategies will change once we have a vaccine and know more about it. But this is a really dynamic system. And so one of the reasons I wanted to sort of emphasize, in the title of the talk being sort of science defining our future, is that we are learning so much about this so rapidly. I can't express to you how much information we have learned in such a short period of time, and what a remarkable scientific and public health focus has been put on this. I've never seen anything like it. Don't know of anything in history that's been like this. And so we're really at a new place in which public health policy and the science behind that policy is so critical. And one of the reasons that this is going to be super important is that public health funding in general over the last 15 to 20 years has declined. And because of that, we are not as prepared today as we might have been otherwise. And this is a long-term thing. This isn't a recent thing. It's because we've sort of become a little bit blasé and that we can solve all these problems. Well, we need to think about what public health looks like under these conditions. And I think this has opened our eyes up to both sort of public health from a healthcare perspective, a policy perspective, an economic perspective. And today I would say that economic factors are part of public health, and those wouldn't have been things I would have thought about before. And so really the science of what we're learning, and then the policy that we're generating and thinking about that public aspect of it, to me, is what really is going to be the focus going forward. And then hopefully we will have a vaccine soon enough so that we could have this lecture live. So I'll go ahead and end it there, and I'm happy to answer any questions that I can.
There were definitely some questions. I am going to invite people now, Stephen, that you have finished. If people still have questions that were not answered up till now, definitely feel free to unmute yourself, and you can ask those. You can also put them in the chat as well.
This is Martha, actually her husband.
Scott.
A very simple question. It may be a little too detailed, but with the messenger RNA viruses that are lipid encapsulated, do you believe there's a risk of inducing autoimmune problems with vaccines like that, like Moderna's?
I think it is very low. There's actually quite a history from gene therapy on forward on using these kinds of particles. And as a result, there isn't any great evidence to suggest that. However, there may be some people that would be susceptible, and they would be susceptible anyway, that may have some of these alternative responses. And so we have to look at those and really monitor them quite well. But at the same time, the risk, I think, within the population is very, very low. And it wouldn't be something in which I would expect to see in the population at large. There's no data that would support that right now. And in fact, it wouldn't have made it past phase 2 trials if there was any indication of that.
Why do you think the FDA has not approved any messenger RNA vaccines today?
Because it's a new technology platform, and messenger RNAs and lipid particles have been used in some gene therapy processes. But one of the things is when you have a new, what we call platform, the FDA is very, very cautious about saying, let's use that as a new therapy until we can prove it. So one of the things that's happened is that because we needed to take new approaches, it gave the opportunity for new platforms. There's a number of them out there to be tested, and that we're looking at them in a very different way. Under normal circumstances, you would have to very slowly and methodically go through and prove that they will do what you say they're going to do without causing harm. And so as a result, it's just, we've only had the technology to do that for about a decade, and it's taken that long to get it to the stage where we can do things. And it's mostly because it's been very methodically studied. this has been sort of an area where we needed to advance things quickly and take any good idea that has that kind of substantial promise and move it forward. And so that's why we're seeing it sort of show up when you hadn't really heard about it before.
Thank you.
Hi, Stephen. This is Jess.
Hi, Jess. How you doing?
Good.
Great.
The cytokine storm that we see in patients, in some patients anyway, is that less likely to happen in people who have weak immune systems?
I don't know that I've seen any data that would specifically address that. I think it would really depend on why their immune system is weak. Because oftentimes weak immune systems are not entirely weak, but they're unbalanced. And if they were unbalanced in such a way that it would promote the cytokine storm, then that would be a problem and they would have even less chance of fighting it off. If they were weak because they had fewer components of the immune system, that puts them at risk for all sorts of other issues besides the cytokine storm. And so I don't see any circumstance where that would provide any protection.
Thank you.
Sure.
Are there other questions out there?
I did have another question. I was waiting to make sure nobody else had one. The blood test that was developed for the UK, it sounds as though blood is not necessarily the best.
This is an antibody test, so we're
Fluid
actually
test.
searching for antibodies. We're not searching for the antigen. So the blood isn't a good
Oh,
source
okay.
of the virus, but it is the prime source for the antibodies. So if I didn't make that clear, the blood test that I talked about that we were part of is an antibody test to identify if you have antibodies developed to the virus. It's not looking for the virus itself.
Okay, that makes much more sense then. Thank you.
I have a question if you're not going to do the ones on the chat.
Well, I was hoping you might just chime in or maybe you can read some of those, but please go ahead.
This is a kind of quick question. I was hospitalized over New Year's and I was told I had coronavirus. And at the time they tested me for 17 viruses and I had RSV and coronavirus. And I'm pretty sure it wasn't the new one, but at the time, if you had COVID-19, would it also show up as coronavirus, the human coronavirus?
In general, no.
Okay.
So one of the things that you have to do if you're developing a test for COVID-2 is actually demonstrate that it doesn't recognize SARS-CoV or MERS or any of the other human coronaviruses so that we don't mistake that for a different type of infection or a different type of disease.
Thank you.
And therefore the testing should be specific. Now, if that was tested back in January, it's possible that the specificity in some of those tests had not been fully developed, but I would be very surprised.
Nobody else got it, so I figure that it probably wasn't. But
yeah, I mean, one of the issues is the sort of the timeline of this was prime flu season. It was prime cold season. So there are a lot of different things that can give you the same symptoms,
right.
but be from different sources. And we weren't able to sort a lot of that out early on. Now we can.
Thank you.
Yeah.
Stephen, there is one kind of detailed question here in the chat from Arnie Kurtzner. Is there any research using CRISPR to change parts of the mRNA or DNA genome and develop a changed form of the spike proteins which may elicit an immune response?
Okay, so that's a type of technology that's called CRISPR, and you may have actually seen that show up. It's been used pretty widely now. Not as therapeutically as it is in research, but it's pretty close to being used therapeutically. There are actually some diagnostic tests that use CRISPR. to identify whether the virus RNA is present. And so there are some tests that are using that technology, and I am certain there are people thinking about using that as a therapeutic mechanism. I don't know of any that have advanced that, but as you can imagine, there are so many different things that are being looked at and tested. And so I'll just give you an example. There's a pre-publication site that's called medRxiv or bioRxiv, in which people write up papers and data before they get actually peer reviewed, as a way of getting data out fast and having them be reviewed sort of by the scientific community at large. For COVID, since January, there have been about 7,800 papers that have been published in that archive. It's about 35 a day since we started studying this. And that's only a handful of the amount of studies that are going on. And I know with a lot of the companies that we work with, they've shifted. They may have been working on cancer, but they've actually changed their focus to work on COVID during this time. I've never seen such a dramatic change in focus across the board for any other disease, not even HIV. And as such, I would be extremely surprised if there aren't another number of companies who are using CRISPR in their process that aren't asking that very question. But I do know there's diagnostics that have been developed that use that same technology.
And were there any other questions from anyone before we say goodnight? Well, Stephen, thank you so much for being here and for presenting all this really complex information in a way that makes it understandable for all of us non-scientists. We're deeply appreciative. So thank you so much for being here, and thank you to everyone for coming and being a part of this talk tonight.
Great, thank you.
Thanks
Everybody
for the
be
talk,
safe.
Stephen.
You're welcome.
Yes, everyone be safe, be well, have a great night.